First, you must realize that you have no idea before you can know the idea. We scan across all the frequencies if we want to learn anything new. Let us begin.
What has physics done for me lately? Second more, the equation E is equal mc squared. We have now acquired a fateful power to alter and to destroy nature. That's like when you're in physics and you go to Dreamworld saying, this is a physics excursion. Science, what is it all about? The whole of human history all falls in the dust of one stroke of a nail file. You can't really get to grips with evolution unless you realise what an enormous amount of time our own planet is only a tiny part of the vast cosmic tapestry, a starry fabric of worlds yet untold. You are tuned into what can only be described as the best radio station on this blue dot we call Earth. It is, of course, 4ZZZ, your community broadcaster, be it on your conventional wireless radio by tuning into the classic frequency of 102.1 FM, digital devices such as DAB or smart speaker, listening via the Community Radio Plus app, or streaming us live from our sensational website at 4zzz.org.au. And of course, you can always listen back to us or any 4ZZZ show for that matter using the ingenious on-demand feature also found at that URL. We also now have a weekly podcast of the show for your listening pleasure, curated by Toby, a condensed version of the show without the music, which my mum prefers. Just search for our show name, which is of course No Idea, spelt with a K, your weekly dose of science. And joining me for episode number 666 plus or minus 3 to speak about all things science are two of my favourite science influencers. Good morning, Peter.
We love to have a range, don't we?
Good morning,
the
Gabe.
plus or minus.
Famously, a measurement is useless if you don't report its error as well.
Yeah, we love having a little error rating. That's what we're here
error
for.
is what I did last week by calling it episode number 666. We haven't tracked the numbers in about 2 years.
So who knows?
That's right.
And I thought I'd done the calculation right. I just tried to calculate the number of Wednesdays there'd been since our last time we knew what the number was.
Yeah.
And then after the show, Toby, who edits up the podcast, went, no, it was number 668.
So we actually... And was the show too before that quite cursed? Because I remember it being quite cursed. So maybe it was cursed without us actually knowing it.
That's right.
I think that's the best way to go through
well
cursed
said.
shows is unknowingly. So this is show number 669. I'm sure Issy will be very happy about that. A semi-prime number, because it's divisible by 2 primes, 3 and 223. Coming up on the show. I've got an update on some more of this chimp alcohol consumption research.
Cheap dates.
Another one. We've got 3 studies in the year. How good is that?
Chimps are absolutely slozzled.
I'm loving this. We'll get into. Well, that's exactly what they've done. Yeah. This is the next step.
Oh, they've gotten them swazzled.
How swazzled do the chimps get?
We had this question. We did have this question. We didn't know.
So this is what they've got into in this recent stuff. And then I'm going to do a quick run through of the 2025 Nobel Prizes a little later on as well.
Yep.
What do you got, Peter?
I've got some stuff on dolphins, and then I've got a brand new day, maybe.
Max?
I've got a mice study. So we'll see how that turns out.
Oh, classic.
To do with cancer.
Is that part of the
Absolute
MotoRap, Max?
Hey?
Is that part of the MotoRap?
Yes, it could be. Did you watch the race on the weekend?
Of course.
Yeah.
And we'll get into that as well.
We will. Okay.
Unfortunately.
I'm assuming you got some weird science, Peter.
I actually have one of the craziest, weirdest findings I've ever heard in marine science.
Huge.
We're going big today. We might... I can't believe I'm saying this. We might finally have an idea of one of the reasons why dolphins and whales beach themselves.
Really?
And it's nothing I would have expected.
Russian subs.
That's it, Max. How did you guess it? Man, wow, you're just so on it. All right, this is going to be a long one, and I'm going to need everyone to lock in, okay? Because it's complicated. So when dolphins beach themselves, people work really hard to get them back in the water, but often it's not enough. The individual dies. And when that happens, if there's a lab nearby, marine biologists will often do an autopsy on the animal.
Yeah.
It's one of the ways that we learn more about these animals. I mean, obviously it's a biased data set 'cause they've all beached themselves, but you can find out a lot from this sort of stuff, like what sort of, diseases they're being exposed to, all sorts of things like that, genetics. So this team of researchers in Florida, and I don't have a QS ranking for you because they're from a bunch of different interesting institutions. A bunch of researchers in Florida in the US US looked at the brains of 20 common bottlenose dolphins that were found stranded in the Indian River Lagoon. Now, this is a pretty big estuary on the east coast of Florida in the US, but the key thing about it is that it has frequent occurrences of harmful algal blooms. So we'll be pretty familiar with that now because we have one happening in South Australia at the moment. To be clear, not all harmful algal blooms are the same species. There's many different species that are a part of harmful algal blooms. A lot of them will be a cyanobacteria. A lot of them will have neurotoxins. So that's a common thing, but it's by no means all are like this. But neurotoxins are very big that's what's happening in South Australia. That is also one that has a neurotoxin. It affects the brain. So anyway, one of the things in this place, in the Indian River Lagoon, is that it also, the increase in frequency of the harmful algal blooms in that area have been linked to climate change. They've been increasing. It's linked to the warmer water that has been coming through climate change. Apparently, the water in this area has been increasing. It's like the increasing temperature at such a rate, like it's a hotspot for ocean heating in this area.
Quite literally.
Yeah, genuine hot spot. So they looked at the brain tissue from the dolphins, from these 20 dolphins that they collected across different seasons, and they compared individuals from bloom and non-bloom periods. Then they did a chemical analysis of the brain samples to count the concentrations of 2,4-diaminobutyric acid, or we're just going to call that DAB from now on.
Okay.
And aminoethylglycine. You don't really know about those. It's just DAB we're using, which are these toxins that come from harmful algal blooms. Okay,
Yep.
So that's the thing that we're looking for, is DAB comes from the algal blooms. Then they looked at the brains and they did histological staining and immunohistochemistry to identify beta amyloid plaques and hyperphosphorylated tow tangles. Now, from those two things, can either of you guess where I'm heading from and what the researchers are looking at?
Give me those words again.
I will. Lots of people listening will know this. They'll know what these two things are. Beta amyloid plaques and hyperphosphorylated tow tangles, or TAU. I think it's tow.
Are they getting early onset Alzheimer's?
Is that Alzheimer's, beta amyloid plaques?
Alzheimer's.
Yeah.
Beta amyloid plaques.
Once you said plaques.
Yeah, you got it, right?
Yeah.
They're looking for hallmark features of Alzheimer's pathology in humans. So that's like part of the disease in humans. We look for these two things.
Right.
So they got to this. It seems like a huge leap, but they got to this because of two things that we already know. Number one, they already knew that in Guam, which is quite, close by, people with Alzheimer's disease are more likely to be exposed to cyanobacterial toxins. So the toxins from harmful algal blooms, or similar toxins to harmful algal blooms. So they knew that people, humans with Alzheimer's disease, are more likely to be exposed to those toxins. And they also knew that dolphins, and I didn't know this, dolphins get a disease which looks a hell of a lot like Alzheimer's. They're actually already used as what's called a natural model for the disease because as they age, they develop those two main neuropathological changes, the amyloid beta plaques and the tow neurofibrillary tangles. So those two things are both in dolphins and humans, so we already knew that. They already were looking at dolphins for how they develop Alzheimer's as like a way of figuring out whether we have any similarities, trying to cure the disease. So essentially, the researchers thought to themselves, maybe they're stranding themselves because they're being exposed to all these harmful algal blooms or eating loads of fish that have, and it's causing them to develop Alzheimer's. That is a huge leap to me. This is why I'm saying this is one of the weirdest things I've ever heard, because it's a crazy linkage of different things coming together.
Yeah.
And it looks like they were right. This was published in Nature. This is no small deal. This is like very heavily researched. They looked through everything. They've close to proven the link here. This is very, very good science. So to put that all together, they found dolphins who had higher exposure to harmful algal blooms, had very different brain chemistry, with increased Alzheimer's-like neuropathological changes. Essentially, these dolphins may have beached themselves because they had Alzheimer's and had gone on a wander, like humans do. Like humans with Alzheimer's will often go on walks without, it's called a wander or wandering. You don't know where you're going, you're just walking, you're just moving, and they often end up in places where you shouldn't be, and it's dangerous. Like people have been known to walk into streets, right? They think it might be a similar thing in dolphins, that they're just wandering because they have an Alzheimer's-like disease. One really interesting thing that I want to point out here is that 5 of the 20 dolphins they looked at were actually juveniles. So that I couldn't see much in the paper. It was very dense, to be clear, so I didn't really digest all of it. But I couldn't see anything talking about the differences between the juveniles and the adults, but I'm sure they talked about it at some point. But let's just get into the results. So all of the dolphins had DAB, that toxin, in their brain tissue, but dolphins that were exposed during cyanobacterial bloom periods showed up to 2,900 times more of it. Huge difference between those that were like
during
more
the bloom
during
periods.
the bloom periods. They all affected dolphins had beta amyloid plaques and toe tangles, mirroring that Alzheimer's pathology. And then they also looked at genes. I didn't really talk about this because the genes thing is really interesting. So they found, I think it was like over 500 different genes that were mirrored in dolphins to humans where their expression affects Alzheimer's pathology, right? And then of those, they found 63 genes whose expression correlated directly with the levels of DAB, so that cyanobacteria. The more of that toxin you have, the more or less these genes are expressed, right? So maybe one of them affects the amount of plaque in your brain or your blood-brain barrier or things like that.
Yeah,
right?
they've pinpointed genes within the dolphin genome that are linked to, if they have them, higher levels of this toxin.
And in the human genome. So they're mirrored. Okay. That's the main thing. And yeah, they found that 15 of those overlapped, right? So essentially it indicates that cumulative and seasonal modulation of these genes, so the more of this harmful algal blooms they're in, the more often, the worse it gets. So that's great. They also found that the dolphins that were in the Indian River Lagoon, like this one specific area of the lagoon that was more repeatedly exposed to the runoff that had cyanobacteria in it, were worse. So the more you have this, the worse, just really layering on the evidence here. And weirdly, side note, they also found that some of the dolphins appeared to have experienced hearing loss, which is a known risk factor for the disease in humans. It's like one of the things they look for neurodegenerative diseases, not just Alzheimer's, but any neurodegenerative disease, they look for hearing loss.
And if you're an animal that relies on sonar, that's probably a pretty big deal.
Probably a pretty big deal. If you and your pod are going somewhere and you can't hear them, not helpful. So yeah, this study is the strongest evidence yet that a chronic exposure to cyanobacterial toxins such as BMAA and DAB can lead to neurodegenerative disease in wild mammals, and probably also us. It demonstrates that dolphins' navigation errors and strandings might be from this cognitive decline, and it's showing like the bioindicators that toxic algal blooms are changing brain chemistry of marine mammals. So this has a lot of implications. Number one, for marine conservation, obviously it's huge because we're seeing now that these harmful algal blooms that are increasing because of climate change don't just affect things in the now. They can cause complex neurological diseases that affect them in the long term. So it's not just a can you survive through the bloom once, it's can you survive with the toxins it puts into your system. And to be clear, one of the reasons they're looking at dolphins for this is because these toxins bioaccumulate, which means if I'm a plankton and I have it in my system, when a fish eats me, all of those toxins go into the fish. And when another fish eats that fish, all of those toxins go into that fish. And so dolphins can get it just through their food. They don't even need to necessarily be in the bloom. So they also think this is a huge warning for public health, and they're thinking like, hey, for people who are living near these toxic algal blooms, it can be it's just something you need to think about, is that it can be a risk factor, which we already knew to an extent, but it's really like hammering home that pathway. And it's also interesting for Alzheimer's research because it strengthens the case that it's actually not just genetics that are playing a role in this disease. It's your environmental factors as well.
Yeah.
Cool.
I think it's interesting that they still found beached dolphins in the same area outside of the bloom period. So they're not at all suggesting that this is the only reason that these dolphins are beached. They're sort of
saying it's the only reason.
These algal blooms seem to be contributing to some sort of neurological disorder.
Disease. They call it a disease.
In the dolphins, which may be linked to the strandings, because during algal blooms, the dolphins that were stranded had really high levels. And all of the
dolphins they looked at were stranded. That's how they got the brains. So you need to be aware that that's where that's coming from.
But
you
also
have
they
a
had
bias.
some stranded outside of algal blooms with relatively lower levels of the toxins in their system.
Yes.
So there's not at all suggesting that it's the only reason that dolphins would beach or any mammal would beach, but a big indication. And I'm sure something that will now feed into, hopefully, any of those biopsies that get done in future strandings for
it should be a
marine
regular
mammals.
part of the autopsy of marine strandings is to figure out, is this one of the reasons?
What do you got, Max?
Today, it's the third Wednesday of October. So apparently it can only mean one thing. It just happens to be Weet-Bix Day.
What?
Yeah, I know.
What are you talking about? Sorry.
The third Wednesday
mean?
of October is
bizarre.
Weet-Bix Day.
When did they decide that?
I know.
And why isn't it like a date? Why is it just
There's no way.
third Wednesday?
There's no way you filled a playlist with this, Max.
Wait, really? No, there can't be that many songs.
So to celebrate this classic breakfast, why not some songs featuring the word breakfast?
Oh.
The year is 1979, and a popular pub band releases their much-anticipated second album, Breakfast at Sweethearts. Sweethearts just happens to refer to a popular cafe in Sydney's King Cross, which is no longer. It has subsequently been replaced by McDonald's. It was a simpler time back then. So on this day, let's celebrate Cold Chisel's second album, Breakfast at Sweethearts, with their title song, Enjoy.
Biological representation in emojis is a joke. I'm sick of it. Keep looking for stuff. There's like 4 versions of a chicken and 1 eagle, and then there's no other bird.
Okay, but we do need to acknowledge our own privilege in this situation, is that Japanese people love Australian animals, and so there is a koala and a kangaroo on there. Like, we're slightly overrepresented.
And 2 types of mushrooms.
Compare that to New Zealand, there's no kiwi.
There's only like 2 trees. One of them's a sapling.
Hey, there's a palm tree.
We just heard from Cold Chisel with their song Breakfast at Sweethearts. You tune into 4ZZZ, show's No Idea, and it's part 2 of this. Do you like your emoji sting, Gabe?
Oh, I stand by that, every bit of it, because it's still crap. The taxonomical representations of life within the emojis is rubbish.
Can I just point out that we only got skin tones, 5 years ago?
I know.
We might not get, rainbow lizards or something that you want until, give them time.
Amoebas, where are they?
True.
Where are the tardigrades? That's what I want to know.
What do you got for us, Gabe?
Max, I've got for you what I think has turned into our favourite side quest science throughout the year.
This year, yeah.
Building off the back, I was going to get into this earlier, but with Dr. Jane Goodall passing away, I did a piece dedicated to her and sort of flagged in that we had an update on this story about the alcohol consumption of chimpanzees, which has... Like we said,
chimps.
This is the third big paper about this year.
Yeah.
What? And,
Is it all the same research group? Are they just putting it out, or is it?
It's a lot of the same people, but there are different people involved and different parts of Africa with different populations of chimps. So it isn't just one specific thing. And it is starting now. Like, this is new research. It's not like there's been this happening before. It goes back to 2004. Where the concept came up, and then there was a book about it written by what's his name?
Some dude. Some dude.
Yeah,
some guy wrote a book.
He said professor. Is it biology professor at University of California, Berkeley? I will get his name. But he wrote a book in 2014 on it and gave it a name. Which, you ready for this?
Yes.
The drunken monkey hypothesis.
Oh, cool.
And the apes too. I mean, who's what? But I guess the monkeys do it too.
Hang on. Whoa, I rescind my clap. Sorry. Wrong.
So what we've talked about previously is first a study came out early in this year about chimpanzees in Guinea-Bissau eating breadfruits with alcohol content.
Yes, but it
off
was
the
low.
ground.
It was a low alcohol content.
It was, but the breadfruits were large. So the breadfruits themselves, max 0.6% ethanol content in the fruit. But they're very big. They're big jackfruit-looking things.
Like eating 2 liters of kombucha.
And so that was the first study. And then the second study went into what they called scrumping, a new type of foraging behavior.
That was cool, eh?
Which was where they looked at which of the great apes are out there actively eating ethanol-rich fruits, which is still very low percentages compared to like what we'd have in drinking alcohol.
We have a much higher tolerance.
High in terms of anything else that we eat naturally. And what they found was that the 2 types of gorillas, so mountain gorillas and western gorillas, chimpanzees, bonobos, and humans all actively eat ethanol-rich fruits in natural behaviors. And so that's scrumping. They went back through and they gave it a name of picking up these fermenting fruits off the ground. But the orangutans don't, which lends more evidence to this idea. Somewhere about a little over 10 million years ago, we had a last common ancestor between us, the gorillas, the chimps, and the bonobos, where a single amino acid mutation changed a single amino acid expression, which then allowed us to process ethanol properly, which means it unlocked this resource of ethanol-rich fruit. The things that were further back in our evolutionary tree, like the orangutans, didn't get that, and so they don't do it. And what the question has been, though, from a lot of this stuff is, how much alcohol are the chimps actually consuming?
Yeah, and the other question we had when we were talking about this was not just how much are you consuming, but how much are they feeling it?
And how much are they feeling
Because
it?
it's like, obviously from human to human, there are different tolerances, but we know that humans have a tolerance for it. So
this new study has the answer to how much they're consuming. We know how much is being consumed by one chimp community in Western Africa and one chimp community in Eastern Africa. Maybe we won't go to the whole of chimps.
It's a good study.
So this comes from researchers across the USA, Cote d'Ivoire, and France, but was led out of the University of California, Berkeley. Can I get a QS ranking?
Oh, USC is high.
21.
34.
University of California is 17 in Berkeley campus.
Yeah, Berkeley's really high.
So in this research, they looked at 200 fruits from about 20 different fruiting trees that the chimpanzees like to visit to eat.
Any good ones?
And they included something called a guinea plum, a bitterbark cherry, which apparently tastes a bit like banana bread.
Oh.
African mulberry, white star apples, calabash nutmeg, and a bunch of other stuff, including
sounds delicious.
various berries. What they found was that the alcohol content, they went and did really in-depth studies of the alcohol content within all of these fruits. And what they found was that there's huge variability. Some of the fruit has virtually zero or actually zero alcohol content, but others, like figs and plums, tend to have very high alcohol content. So big variability in what they're eating. On average, of all they looked at all of these trees that they know they eat from at least a bit. On average, the alcohol percentage was 0.26%.
Low. Very low.
Low. But on par with what they found with that breadfruit too in the other study.
Yeah.
However, there's a catch, because they also spent time figuring out which fruits the chimpanzees feed on the most. Which fruits they're actually spending their time going at more than the others. And if you actually calculate the average percentage of alcohol of the fruit they eat, it's higher than 0.26%. The chimps actually seem to be having a preference and seeking out the fruits with higher ethanol content, which included the figs and a couple of others like those bitterbark cherries and the guinea plums.
That sounds nice. If alcoholic banana bread, someone should be working on this right now. Where is the banana bread liqueur? What are you doing?
So the average alcohol content of the fruit that they're actually eating in these two chimp communities is higher than the average across all of the fruit. But what does that mean overall? Okay, let's break down the numbers.
Yes.
The study says chimps eat an average of 4.5 kilograms of fruit per day.
Oh, that's like your dream.
So if they have this average rate, I think it was in the 0.3 something percent. It varied in the different communities. It's in that range of what alcohol content is in sort of 0.3, 0.4%. It equates to over 4.5 kilograms of fruit, 14 grams of ethanol. Now, in Australia, we use the international standard for standard drinks, which is a 10-gram standard. Okay?
Oh.
So 14 grams of ethanol is 1.4 Australian standards in an average 4.4 kilogram day of fruit for a chimpanzee.
It's having like a glass of wine with dinner every night, basically.
It's, 1.4. For example, they said, ingesting 75 ripe figs in a single feeding bout, which is something they propose is what they do.
That's crazy.
And those figs have about 0.5% ethanol content in some of them. That would be a 10-gram standard drink worth of ethanol. In 75 figs. The thing to keep in mind, though, is chimpanzees weigh a lot less than we do.
Yes.
They're about 40 kilos on average. Very variable, of
they're
course.
also
like
eating while they're drinking. Right? When we think about alcohol content, we're not actually consuming any calories along with it. Oh, great.
There's so they're 40 kilograms on average. So
little babies.
Max, you and I have established before we're about 3 foot high, so we're about the same as the chimps.
Right.
Are in that 40 kilo rate. But for a standard
human,
a biological male human, let's say 80 to 90 kilos, about double the chimp.
Yeah.
So the chimp having the 14 grams of ethanol is more like that human male having 28 grams of ethanol, like kind of double that amount. Right. So because
it's like having 2 glasses of wine with dinner every night.
They've got less to them. There's less blood for that alcohol to get spread around. There's less cells to metabolize that alcohol when it's in their system. Potentially, it has a bigger impact. A standard drink in a chimp might be 5 grams or something. Assuming that it affects them in a similar way that it affects us.
You need to think about the body composition as well. When I was studying blood alcohol, I remember we had to make an equation or something to study blood alcohol, and I had to add more for me because people like me, women, will have higher fat contents. So
that 14 grams of ethanol per day maybe is more like 2.5 chimpanzee standards if they have a similar sort of metabolism and impact that we do. So on the other hand, like you said, Peter, pushing it the other way into sort of less drunk territory, they're getting this ethanol, this alcohol from a high level of mainly food and a bit of drink. There's some chimps that actually will go and there's one community that will go and get alcoholic sap that humans are tapping and drink it with leaves. But even in those cases, even those extreme cases where they're scoffing alcoholic figs or stealing human alcoholic sap, they're having so much food that they think their stomach would possibly fill up before it gets to a point where it would be a level that would make a human drunk in the equivalent sort of amount of alcohol, which is why we probably don't see chimpanzees stumbling around the forest after hitting the figs.
Is it ethical to just offer one a drink? if they're really trying, maybe it's ethical to just offer a gift and just see, hey, how do you feel?
So it's basically saying we think chimpanzees maybe get buzzed off figs and plums and other things, maybe get tipsy on an extreme example, but probably aren't getting drunk to the point where it's going to significantly, and there's evolutionary drivers behind that, right? You don't want to be stumbling around, not being able to escape and hunt and all that sort of stuff. But it is further evidence behind the drunken monkey hypothesis, pointing to the evolutionary importance in how a common ancestor between us and chimps and bonobos and gorillas some 10 million years ago may have led to an evolutionary advantage from suddenly being able to consume ethanol. It may have helped us smell and taste more ripe fruits with higher energy content. It may have allowed us to come down from the trees at a time when the African forest was shrinking, that 10 million years ago, and eat those fermenting fruits on the ground, which is one of the interesting. That was something that wasn't actually substantiated by this study because they found the average ethanol content in the fruits, at least in today's age, was the same in the canopy versus on the ground of what the chimps were eating.
Yeah, we need to figure out what the fruit was like 10 million years ago.
So maybe when this was evolved, there was higher ethanol content on the ground. Maybe there's more study needed, but they didn't find a difference in the trees and on the ground. And like we've talked about previously, it could unlock things like social feasting behaviors that have then spread into cultures that have ended up in humans across the world in all sorts of different ways. And the really interesting other side of the drunken monkey hypothesis on the serious level is that this evolutionary advantage from identifying or exploiting ethanol-rich fruit help us explain alcohol abuse in modern humans as sort of a side effect in certain instances.
Because your brain is hardwired to search it out.
Because we maybe have this evolutionary driver to search out ethanol-rich fruit and exploit that resource from the forest floor way back in our evolutionary history that might be related to why modern humans also can have alcohol abuse.
Can I suggest something for in between now and next week?
Go for it.
Anyone who knows you knows that you love fruit.
You want me to try and hit 75 things in one go?
I genuinely think if anyone can, you can. And it's your dream. It's your dream. Think about it. You can do it for science.
To do it fairly, though, I'd need to double what the chimp was eating. I'd need 150 figs to get the equivalent amount of alcohol.
I think you could do it. Let's find a fruit that's healthy, that's fine, and just try.
Well, if you don't hear from me next week, you know why. What do you got next?
Now, my study, before I start, I should make it abundantly clear that this is a mice study or mouse study. No humans were used during this research.
Oh, no. Used. That's good.
Don't get that excited. Researchers at the University of Massachusetts, Amherst, QS ranking.
Oh, University of Massachusetts.
55.
87.
99.
250 247
Well, they got in our heads.
have shown that their nanoparticle-based vaccine can successfully prevent several aggressive cancers in mice, citing the big three: melanoma, pancreatic cancer, and triple-negative breast cancer. In one experiment, the researchers combined their nanoparticle system with well-studied melanoma peptides, similar to how a flu shot typically contains parts of inactivated flu virus. Their formulation was then used to train the mouse's T cells to detect and destroy melanoma cells. 3 weeks later, they tested the vaccinated mice against melanoma. So some good news here. 80% of the mice that received the customized nanoparticle vaccine remain tumour-free. Not only did they resist melanoma cancer, they went on to survive the entire study period of 250 days. In contrast, and a bit of a content warning here, all of the mice that received traditional vaccines or no vaccine, sad face, developed tumours and died within 35 days. It should be noted that the vaccine also stopped cancer from spreading. When the mice were systematically exposed to melanoma cells to mimic the spread of tumours, none of the nanoparticle vaccinated mice developed lung tumours or tumours anywhere else in the body. Meanwhile, every other mouse exhibited tumour spread. So there you go. The researchers...
I was just going to say it's crazy. I was also thinking, I feel like I've read this. Was this about them, like the delivery of the drug?
Correct,
like, was that what
yes.
they worked on? Yeah, it's
Yeah.
about getting it to the right place, which is something that a lot of cancer research is looking into. So it's not just like at the moment, most cancer drugs are just flood your entire body with something and hope to hit the things that you need to hit.
Yes.
So a lot of research at the moment is how can we get it to the right place and not hurt the rest of your body?
So the researchers say that their unique nanoparticle vaccine offers a promising platform that could be used across multiple cancer types. They envisage that such a platform can be applied to create both therapeutic and preventative regimes, particularly for individuals at high risk for cancer. When it comes to its use on humans, the vaccine will probably have to follow a therapeutic approach, treating patients who already have tumours, individuals who will probably die, I'm thinking. And however, the researchers note, even with a successful human therapeutic approach, it will still take years for the vaccine to be FDA approved. So yeah, there you go.
Yeah, huge step forward. Really big, like still super cool achievement. Even if it doesn't work out, it's a huge step forward. Super cool.
And they've created a startup company, and it's called Nanovax Therapeutics, if you want to check that out on the web. You're tuning into 4ZZZ.
They're never going to ask for us back.
It's Max and Issy. And if you want to subscribe, should we just shove that in there too? We want to see some subscribers coming in. Just go to our website at 4ZZZ.org.au/support.
Yeah.
You're tuning into 4ZZZ. We're coming up to our 50th birthday, so we reckon you should subscribe. I remember when I was 50. Good times. What do you reckon, Gabe?
Yeah. I mean, it keeps us on air. The subscribers of 4ZZZ keep us running and keep us bringing science to you every single week. And as a thank you to everyone who does subscribe, you know, you get the 1 minute glow.
You do.
You get 24/7 radio. You get the baby heart
On this
seal
show,
if you do it.
you get the baby heart seal. No other show is giving
Please
you that.
don't expect
Think
that
about
in any other show.
And on top of it all, to sweeten the pot, there's a monthly sub prize you go into the running to win. This month's is Halloween themed, spooky themed.
Yes, it is.
Spooky.
From Berserk, Australia's largest alternative online store, bringing alt clothing, pop culture collectibles, homewares, cosmetics. You go into the running to win a value pack, spooktacular value pack.
Oh, hell yeah.
Valued at $500 with all sorts of ghoulish goodies in there. You can support community radio on 4ZZZ by going to 4ZZZ.org.au /support, and you can look up all of the different subscription options. Max, you got them?
Yeah, so if you're under 18, it's $25. If you're a pet, it's $25. Concession, $40. A full subscription will be $80 for the year. A passionate, $140. Or a super sub, get your name written downstairs on the wall for $500. You can also contact us during business hours. We've got Stephen downstairs at the moment on 07 3252 1555, or you can come and visit us in person at 264 Barry Parade, Fortitude Valley.
And Max, if you do subscribe, your name pops up when you text in. Makes it a lot easier for us to know who we're talking to. And Michael texted in earlier asking what happens if we eat fish on the back of the story about the toxin buildup.
Not just generally.
Algal blooms and the dolphins. yeah, not just generally. But I mean, that's the next bit of research,
it's
right?
It's one of the next things they're looking at is because, like I mentioned, fish, it's a bioaccumulative toxin. One of the ways it accumulates dolphins is through eating fish that have been exposed. And so, yeah, they think that could happen to us too. It is not substantiated or proven. It's one of the things they're going to look into, but it is something that seems maybe likely. I also want to point out here, because we are in Australia, I have seen nothing about the South Australian algal bloom, that it is this type of toxin.
I had a quick search and it's different species of algae they were looking at in that study.
Definitely different species of algae. This one, the toxin they were looking at is across multiple species of algae. It's a lot of them. But I have heard nothing about this being related to the South Australian one at all.
Right.
Yep. So at this point, just go the advice given by the experts in Australia. But it's probably something that we're going to look into for future.
And there have been marine mammals found washed up on the beach.
There have been, yeah.
Maybe stranded or maybe starved or other things going on. Yeah. But there's stuff to look into.
Lots of stuff to look into.
So
you tune into 4ZZZ. The show is No Idea, your weekly dose of science. With me, Max. We've got Issy, we've got Gabe, and we've got Maximillian. So it can only be one thing.
Okay, it's time for the best part of the show. Loosely defined as science, yeah, you already know. Everybody listens to 4ZZZ just to hear us talking about what Butters just did. Subscriptions just keep rolling like the tires on a car. But something tells me that our science curves won't go far. But unlike an engine, I won't keep you in suspension. We're all here to hear him talk, so let's give him attention. You're not ready for when he starts rapping. Gonna hand the mic to Max, and I'm not talking Van Stappen.
Let's do it. Supercars happened on the weekend.
It did. Oh, it was so good, Max.
It's a great race. 54 drivers trying to squeeze into 27 cars, driving around the mountain, which is like a 6.2 kilometre track, which goes up a mountain.
Huge elevation.
Iconic sort of racetrack. Anyway, who would be crowned king of the mountain? It was a race of two halves. They started in the dry, but ended up fighting it out in the wet. And the rain normalised the field, which was good because there was a bit of an engine parity dispute. Team DJR, who drive a Ford team, made a protest on the Saturday saying that the Chevrolet had a slight power advantage over the Ford, but this was dismissed. It's something like 2 kilometres quicker or something per hour quicker. So, but over the course of one lap of 6.2 kilometres, I guess it's significant. Anyway, the race saw the usual number of safety cars along with all too many pit stops for tyres and fuel. But as they say, there can only be one winner. And as it turned out, the car that claimed a line on us was given a 5-second penalty, which meant they ultimately placed third. So second and third finishing cars were elevated to 1 and 2.
Yeah.
It's crazy for such a long race how the gaps are that tight.
Yes, it was amazing, wasn't it?
Yeah.
Now, I cannot tell you, I can't give it a good explanation the whole race. Your best bet is to go and watch the what do you call it, the highlights reel.
Extended highlights.
It's 52 minutes long.
I was going to say, look, I'll be honest, I caught the 18-minute highlights, and even then the hard cuts between each, it was literally just crashes or penalties. And even then I was like, I have no idea what's going on. So having to do the research and watch the entire 52-minute
But if you get that vibe from the highlights, I sat there and watched the whole thing. I'm so glad I did.
Yes.
Because it was like that during that as well. There'd be moments where you're like, oh, look, the car with Craig Lowndes in it is suddenly in fourth. Where did that come from? And then you'd blink and he's down in 14th again. And go, what has happened?
You'd look away for a second and someone's in the wall, or someone's Tokyo drifting around the circuit, and you're like, when did it start raining? When did it stop raining?
And all those at the front of the grid, they somehow all crashed out.
At the back?
At the back.
You couldn't get
brilliant,
you couldn't get too emotionally invested in anyone's story because they just end up at the back at the end.
And so it ended up that Kiwi Matt Payne and veteran Garth Tander driving the number 100 Penrite Ford Mustang. I think they started from 16th or something. Yes. They ended up winning the race. So that was pretty good. And who came second? They started like from 12th or something like that. Check. And
Crazy.
He came from nowhere. He's like lapping everyone at the end of the race 1 second quicker than everyone else to get to second place. So Reynolds it was. Yeah. I can't think of his first name. I want to call him Ryan Reynolds, but it's not.
Dave Reynolds?
Dave Reynolds. Yeah.
Are we going to talk about the beer moment as well?
The beer moment,
Something that I don't think would happen in F1. Supercars start.
I think the FIA might take a dip.
Yes, I was going to say, if the Formula 1 equivalent was finishing off the track and walking off into your yacht,
Kimmy did that.
This one was pretty similar. Chaz Mostert, he crashed out, and then immediately someone on the sidelines handed him a beer. And I was watching, I don't remember who was commentating, but they were like, I'm going to place a bet. He didn't even finish his sentence before a beer was handed to the driver. So if you saw that moment, that made headlines.
They did.
So that was from this race. But yeah, what a fantastic race just to passively watch as well.
Gabe, any more to add, Maxi?
No.
On that?
That's it.
Extreme H happened on the weekend. So this is a follow-on from Extreme E. So it's the next iteration of the carbon neutral rally racing series, and the series has evolved from using just batteries in the Odyssey 21s to the next generation, they call them Frontier 25s, with onboard hydrogen fuel cells generating the electricity required to power the buggy's motors. Fittingly, the inaugural Extreme H race was won by Team Jamil, featuring Australian rally driver Molly Taylor, who also won the inaugural Extreme E back in the day. There are also some new team owners, most notably Australian-based British techno club DJ and radio DJ, just like us, Carl Cox, with his team...
We're really lumping ourselves in with Carl Cox.
With his team elegantly named Carl Cox Motorsport. So maybe Atlassian should buy an Extreme H team for some carbon offsets, I'm thinking. And then IMSA, which is sort of the US equivalent of the FIA WEC.
Of course.
As we get ready to see a new team on the Formula 1 grid in 2026, Team Cadillac has shown some form, winning last weekend's Motul Petit Le Mans. It's an IMSA.
Because it's in America, do you have to pronounce all the things you wouldn't pronounce if you
Yeah, probably. The car was not Ferrari powered. They used their own 5.5 litre double overhead cam V8 engine, but this bodes well for their chance next year when they join Formula 1. And being the last race of the series, we saw number 6 Porsche, co-driven by Aussie Matt Campbell, finish third in the race, which secured both this year's IMSA Drivers and Constructors Championships. Interestingly, Porsche has pulled the pin on racing in the WEC series, hypercar series, after this year, which Ferrari, good news Ferrari will probably win that championship, the WEC, the World Endurance Championship, which is the European version.
At least something good's happening in Ferrari this year.
And then I want to open up the floor for a general discussion on F1. So I'll just kick it off. With the constructors having been decided in McLaren's favour after Singapore, should we now turn our attention to the Drivers' Championship with points available in the 6 remaining races and 3 sprints? So how will it play out for the McLaren drivers? The current standing sees Aussie Oscar Piastri ahead of teammate Lando Norris by 22 points and Max Verstappen lurking another 41 points behind. So, and he's probably the only other driver who could probably upset what I'm calling the Piastri Papaya party. You get 25 points for a win and 18 points for second. Any thoughts? Should they start enforcing some rules?
Well, what were our stats? What were our estimates for wins, Piastri wins?
Well, that's right. Well, Gabe said 8. Maxi said 9, or you said 9 or 10, yeah. And I said 12.
9,
yeah.
So,
I was being a prick to Gabe, clearly.
So where are we? We'll run some data. We'll see how far we are, because that could probably... We'll crunch some numbers and we'll see how close each one of us are.
And how many DNS, I guess, they have to have. Like, if the McLaren start banging
into each other,
banging wheels,
yeah.
and they put themselves out of the race, and then one would assume either George Russell or Max Verstappen will win the race. Georgia's a fair way back, so I don't think he's a contender for number 1 this year. But
what do you think?
Yeah.
Well, I was going to say 6 remaining races with 25 points for a win.
Yeah.
So
and they missed 3.
Yeah, so anything could go at this point. I know constructors has been decided, so now it is just
where the money is,
numbers.
by the way. So does McLaren care if their driver wins the drivers' championship?
I reckon start burning that Papaya rule book.
Gabe, any thoughts on this?
Yeah, I mean, the sponsors will still care if their cars are crashing out 3 laps into a race because the 2 drivers can't keep their cool.
Yeah.
Which is a big, that's the money does talk in F1. But we'll see. Hopefully that we get some good racing for just the third-party viewer, who just wants to see some cars go around quickly and overtaking each other, and they can just sort of have a nice little battle of these 2 orange cars out in the front.
That's right. Well, they've been the pick of the cars, haven't they? Because they finished. Well, they got twice as much as the second place. Who's that? Ferrari or Mercedes or whatever. That's amazing. And are we racing this weekend?
We are.
We are.
At
racing
Coda.
this weekend. At the US. And so that's this weekend. And there's also MotoGP this weekend at Phillip Island.
Right.
The Aussie one.
Oh, sick.
Yeah. So I guess we should continue the discussion next week after we see what happens during this race.
Yeah, how the McLaren pit will decide to manage. Yeah.
Okay, I've got a question from Pete. Loves his MotoGP, but he says, "G'day, legends. Do you think the current F1 cars would get around Bathurst?" Well, we know they don't because they're too close to the ground. And if so, a theoretical lap time.
Yeah. Well, what did Liam Lawson do when he?
He took the Red Bull around, didn't he?
Yeah. Let's have a quick look.
Anyway, we know the supercars get around like 2 minutes 3, 2 minutes 4 seconds. So, would they be under that? You would think so, because there's lots of corners, isn't there?
Pete, I'm going to pose a question. Would supercars be able to drive around Monaco? Because that's what I was thinking this entire time.
I think, absolutely.
I think, because F1 cars struggle to fit. But I was thinking, wow, this race supercars in comparison to Monaco, where nothing moves.
Yes.
Or if it does move, it's because of this big crash and everything has to get shut down. I want to see.
Okay, I got some.
Oh,
sorry,
we got sats. Go. I was going to say, I want to see those supercars on Monaco because that truly would be nothing moving, nothing happening.
And 3 cars
and 3 cars into a corner, yes.
But stuff happens at the GC, right? Like,
true.
I figure that's a pretty tight.
It is pretty tight.
Street track.
Yeah.
I reckon they'd do all right around Monaco. I don't know. Call me crazy. They'd do all right.
We might live to see it. We'll see what happens.
There was a Formula 1 car that Jensen Button put around the Bathurst track, Mount Panorama, in 2011. It was old Formula 1 car, not fully up to scratch. It was really high sprung, so it could get around the track. But they put it in at a lap time of 1 minute 48 seconds. The supercars were doing 2:04s during qualifying.
Yeah, and then Craig Lowndes drove it as well because they were sponsored by Vodafone back then.
Yeah.
So I don't know what time he set, but and then they let Jensen then drove one of the V8 supercars around the track as well, so to see how that went. Yeah, pretty cool. Okay,
cool.
You tuned into 4ZZZ, and the show is No Idea with me, Max. We've got Issy, we've got Gabe, and we've got our friendly neighbourhood marine scientist, so it can only mean one thing.
Do you know what tuna is?
Fish.
It's fish!
Thank God someone's working on this. I don't know what I'd do without it.
Fish are friends, not food.
Great start. Gabe, what does a cat say?
Yeah.
Max, what does a chicken say?
Cluck, cluck.
And Issy, what does a shark say?
You can stop cringing now, Gabe. Where to go first, Peter? You need your mic.
You need a mic. Get that woman a mic.
I'm not there. Oh my God.
That's
Okay.
what happens when you don't show up for the MotoRap. You're not ready for the marine rap.
Why would I show up for them? Anyway, we're going to start today with some tiny things that you guys know live in the ocean. Just hit me with some things that you know live in the ocean that are very small.
On occasion, Max.
Yeah.
I don't know.
True.
Plankton.
Little soldier crabs.
Plankton.
Algae.
Great. Well, today you guys are going to know one more because last Friday was actually maybe the world's first International Coccolithophore Day. And while I'm not usually a fan of capital D days, I'm going to use...
Coccolithophore Day.
Coccolithophore. I'm going to use this as an opportunity to tell you about coccolithophore. So what are coccolithophores. They are single-celled algae that live in like the upper layers of the ocean where there is sunlight, and they live there because they have chlorophyll and they photosynthesize. They produce oxygen like plants. So they're really cool.
Am I correct in thinking that these are the things that produce a lot of calcium that then
you will, and we'll get into that.
Produce very impressive structures?
They produce very impressive structures, even on their own bodies. Now, they're very, very small. They're actually too small to see with the human eye. They are microscopic, but they kind of look like, when you see them eventually, they look like little soccer balls covered in like almost like suckers, like tentacle suckers, just covered in them. They're called calcium carbonate plates. They can be very ornate. The name of those plates is coccoliths. Therefore, coccolithophores. There are many species of coccolithophore, by the way. It's a very big group.
Calcium carbonate's the same thing that's the building block of corals and a lot of shellfish.
Calcium carbonate is everywhere. So they look really, really cool. They're very cool little things, but they do collectively, when put together, cover vast areas of the ocean. I cannot tell you how many of these there are in the world. Like, I don't have a word for it. It's above trillions. There are just so many. And when they die, they sink to the sea floor, eventually forming chalk. That is what chalk is. Chalk is coccolithophores. When I learned this, I was very excited. When you are writing on a blackboard with chalk, you are literally drawing with ancient plankton.
If
How
it's
cool
real
is that?
chalk, I think there's synthetic
If
chalk.
it's real chalk. If it's real chalk. Oh my gosh, just get excited about coccolithophores, man. It's International Coccolithophore Day last week. They also, as Gabe was alluding to, make limestone deposits. So limestone is the stone of coccolithophores, including, if you know them, the White Cliffs of Dover in the UK, those giant cliffs that are bright white. That's all coccolithophores, baby. That's them. That's what they're made of. They're made of ancient marine organisms. And then because of that, they help us with geological records, which I'll get into. So what do they do? Why are we excited about coccolithophores? It's not just because they look cool when they make chalk. It's because they act as natural carbon regulators. Because as Gabe said, they're made of calcium carbonate. They need to get that carbon from somewhere, and they usually get it from the atmosphere, and they draw that in, or from the ocean. They draw that in and make the calcium carbonate. So they sequester carbon through their own bodies. In fact, every single year they produce 1.5 billion tons of calcium carbonate.
What?
Wow.
Insane levels of calcium carbonate.
Yeah.
And then when they die, those coccoliths, the little plates, fall off their bodies, fall down to the bottom of the sea and create those huge deposits. So it literally sequesters carbon, and then the carbon goes into their bodies and it falls to the bottom of the sea. They also do other things as well, like removing carbon dioxide is only one of the crucial ecological functions that they have. The other one is that obviously they photosynthesize, so they release oxygen, which contributes to the planetary oxygen budget. They feed marine food webs. They're pretty much the base of the food chain. There's not much below them, as anything that photosynthesizes and is microscopic, there's not much below you, if anything. They also influence ocean chemistry and nutrient cycles, and also because they're very light colored, they actually reflect sunlight. When the ocean has a lot of them in it, they reflect sunlight back into space and change the Earth's energy balance. These teeny tiny little things. So they're very, very cool. Despite their importance, though, climate change is a threat to them, because of rising sea temperatures, but mainly because of increasing ocean acidity. In more acidic oceans, a lot of animals find it harder to, and not that this is an animal, but a lot of creatures who have to create calcium carbonate struggle to. The more acidic the ocean is, the harder it is to make calcium carbonate, and so things like coccolithophores will find it harder to actually get that carbon into their bodies and make their bodies. So those changes could destabilize ecosystems and reduce the ocean's carbon storage capacity. But here's the thing. There's one more thing they've done. They have also recorded how all of this went down before. Because, as I mentioned, those coccoliths fall to the bottom of the ocean and form large deposits, they actually record the history of the Earth's climate through their own fossilized shells. Like, we literally, if you dig through these deposits, and I've done it in class, genuinely, you can go through as a core of these deposits, and you can see how, like, carbon ions and stuff change over the years, and it gives you an insight into the Earth's climate. So, really cool. They also offer clues into current and future warming. Obviously, we are going lightning pace with our climate change, so very much in new territory, but still very important context clues. So, we're going to get... This is just some of the research that's going on about coccolithophores right now. So at the Lyell Centre in Scotland, they're looking at how coccolithophores' carbon storage capabilities are affected by the acidification, and they're trying to use computer models to predict which of the many species will remain as effective carbon sinks in the future. So which ones are more effective than others in acidic. More tolerant to acids.
Yes.
In Norway, at Norse, they are looking at their life stories. They're trying to figure out, how do they grow? Who eats them? How do they relate to viruses and viral infections? And how those interactions affect the carbon flow throughout the food webs. And they're using ancient DNA in the seafloor sediments to track responses to previous climate shifts. In the Ruda Boskovic Institute in Croatia, the Cocco team, really cool team name. A lot of these guys have really cool team names. It just seems to be a thing about studying weird little microorganisms that you have fun in other ways. They are looking at how bacteria influence the coccolithophore carbon transformation. So not viruses, they're looking at bacteria and how does that relationship work. In the University of Lisbon in Portugal, they're looking at how aerosols and atmospheric dust fertilize the coccolithophores. So as photosynthetic organisms, one of the things they need to do photosynthesis is nutrients. So one of the common ones is like phosphorus and nitrates. And so things like aerosols and atmospheric dust are known to sort of fertilize the ocean. And so, that is one of the things that they're looking at is where these dusts are, how it affects coccolithophores, and they're doing all sorts of stuff to research that. And so the quote from that research lab is that we are connecting tiny chalky organisms to planetary carbon flows, which is very cool. And then at the International Nanoplankton Association, they are connecting living coccolithophores to their fossil records and trying to sort of date where different species are, how they do, like they're just filling out that record so we have a better idea. And so all of those great institutions that I mentioned have come together to ask the world to please establish October 10 as want to raise awareness of the vital role that coccolithophores play in regulating the Earth's climate and supporting marine ecosystems. I thought we could play a little game here. Guess the highest QS ranking of the institutions that are researching coccolithophores. So we have the Ruda Boskovic Institute in Croatia, the Lyell Centre at Heriot-Watt University in Edinburgh, the Norwegian Research Centre, the Marine and Environmental Science Centre at the University of Lisbon in Portugal, and the International Nanoplankton Association.
I reckon it's got to be Uni Lisbon. I reckon they're 80.
Okay. Anyone else?
I'm going 200 would be the lowest.
I'm going 100.
It is, in fact, the University of Lisbon as the highest of them. 3 of them did not have QS rankings. Between Heriot-Watt in Edinburgh and the University of Lisbon is at 230. Heriot-Watt is 256.
I said 200.
You said 200.
Yeah.
So, I wanted to see, guys, why do we think? Should International Coccolithophore Day be a thing?
If Weet-Bix Day is a thing, why not? Bring it on.
I agree. I also do think someone should probably just tell them that you can buy these things.
True.
Just buy them. There's calendars online, you just pay for it. So maybe just pay for it. But I'm all for it. I'm all for International Coccolithophore Day.
What do the next 50 years of 4ZZZ sound like? We have no idea.
Yeah.
What do you got for us, Gabe?
Max, we got to get into some of these Nobel Prizes because we've got all of them now. The Nobel Prizes for 2025 have been announced. And I want to get into... There's one in physics, chemistry, physiology and medicine, literature, peace and economics. 6 categories. Physiology, medicine is one. And so we're going to just dive into the science behind physics, chemistry and physiology and medicine. So that the natural sciences one. We will leave literature, peace and economics to the other shows. Here's what you need to know about these ones. And there's an Australian in the chemistry prize, which Issy's going to get into. So to start with physics. This was awarded to John Clark.
What is it all about?
Exactly right. Awarded to John Clark, Michael H. Deveray and John M. Martinez for the, quote, the discovery of macroscopic quantum mechanical tunneling and energy quantization in an electric circuit.
I have seen so much about this. People have been freaking out about this online, from what I've seen, everywhere.
So this is like so many Nobel Prizes. This is an award that has been earned for work that actually came out quite a while ago. It came out in the 1980s. And what it's looking at are quantum mechanical effects at the macro scale. So at the micro scale, if you look at electrons, photons, atoms, there's this thing called quantum tunneling. To overly simplify it, the analogy people use is throwing a ball against a wall. You expect it to bounce back. What can happen when something like an electron comes close to a very small barrier is that it can just go through it. Because the...
sometimes they're a wave.
Because the position of the electron has a wave distribution, which means there's a probability distribution, which means it can, as it gets close to the wall, that probability wave can get to the other side of the wall and it can appear on the other side of the wall. All right, we're going to do very big oversimplification.
You're just going to have to, this is the thing with chemistry. When I did chemistry, one of my favorite things about it is that sometimes you just have to sit there and go, yeah, all right, sure.
And so what they were looking at is, well, can this happen on the macro level? Maybe not Schrodinger's cat level scale.
Yeah, maybe not the whole cat.
But at a bigger scale. So what
whisker.
they did is they had a superconductor, series of experiments, but the core one was a superconductor. So you think of it of just a wire with a break in it, cooled to just above absolute zero, so negative 273 degrees, just above Kelvin.
Hell yeah.
And in the middle of that is that break, a thin layer of non-conductive material, something to stop the electrons passing through. And so what they know is when the electrons pass through, you can get a current detectable on the other side. What they were looking for was a voltage on the other side, which would imply it's not just microscopic quantum level stuff happening of electrons jumping.
They've actually jumped a real gap.
Billions of paired electrons passing a voltage through this barrier in the superconductor.
Insulator,
Yeah, exactly. And so what they found is that in this very specific setup that they had, which had a lot of other stuff going on, they could detect a voltage at a certain threshold. They were able to detect a voltage on the other side, which implies this quantum tunneling behavior can occur at what they call a macro level. Still, we're still talking about voltage.
We can't see it, but it's there.
Superconductor. But that's what it means. And
pretty macro for an electron.
It has paved the way for all sorts of advances in quantum computers and quantum technology. It's really important as we get into the development of the chips that are powering the world today, getting smaller and smaller. These quantum level effects actually start happening, and you start getting electrons jumping
just
and
leaving, just going AWOL.
So it's really important, and it comes off the back of this being declared by the UN the International Year of the Quantum. There are some other breakthroughs that were thought may have been in the running for this year's prize, may have gotten pushed out because this is the year of the quantum. It's exactly 100 years after Werner Heisenberg, who is a controversial figure for involvements during World War II.
Yes.
But prior to that, in 1925, published what was very foundational work on quantum mechanics, which we don't need to go into, but this is now 100 years of that. So it was declared the International Year of Quantum by the UN. And so the Nobel Prize probably had a bit of a push towards another quantum breakthrough. Now, the Physiology and Medicine Prize, this went to Mary E. Brunkow, Fred Ramsdale, and Shimon Sakaguchi for their discoveries, quote, their discoveries concerning peripheral immune tolerance. Our immune system, we're going to very briefly get into our immune system. Every day there's thousands of microbes trying to get in, cancers forming that our immune system is exactly right, that our immune system is able to usually deal with, happening constantly throughout your body. And one of the core parts of that is the T cells that we use. One of the core parts of our immune system are T cells, which are able to identify these things like foreign bodies, microbes, and pathogens, right, and cancer cells as well. But the question, there was a big question about how the body is able to differentiate between what it should attack and what it shouldn't attack, which obviously goes wrong. That's what cancer is, right? It's when it misidentifies something as being from your body that needs to be attacked when it's not. There's still a huge amount going on within your immune system to identify what is something I should attack and what is something I shouldn't attack. And so this research is actually a bit of a back and forth between these researchers. Really cool bit of collaboration in science. The first breakthrough was in 1995 with Sakaguchi, who identified, at the time, people thought there was this idea called central immune tolerance, which is like this buildup of all the harmful stuff that would, in your immune system, that would attack the body, got eliminated in the thymus, which is a gland in the chest. Which is very important for immune system and T cells. So they thought all that stuff was just sort of getting eliminated by the thymus over time. You just build up your thymus was getting rid of it. He was like, he showed through some work that it was more complex than that, and there were things that we now call regulatory T cells that come in on site and sort of are policing forces of what gets attacked and what doesn't. We won't get into the specifics of what's going on, but found this breakthrough in T cells. In 2001, the other two researchers, Brunkow and Ramsdale, made another key discovery when they found that an explanation for why a specific strain of mice was particularly vulnerable to autoimmune diseases, which is a mutation in a gene they called FOXP3. 2 years after that, Sakaguchi, the original guy, was able to link these two discoveries together, showing that FOXP3 gene governs the development of the cells he identified in 1995, now known as regulatory T cells. And so it was this big breakthrough that allowed us to understand so much more about how the immune system identifies what it's and deals with what it isn't going to attack and what it shouldn't attack, and how it does that cleanup effort, how it does that policing.
Basically, the bureaucracy of your immune system.
Yeah, exactly right. And it's been really important for our understanding of how the immune system functions, and is now, there's a lot of sort of early clinical trials that are built on the back of this, looking into all sorts of immune responses, including cancer work, that is based on this discovery of these regulatory T cells. And that is the Physiology and Medicine Prize. Izzy, you want to do the Chemistry Prize?
Yeah.
I love chemistry.
This one was the one making headlines because it featured an Aussie, Professor Richard Robinson, with one of the researchers taking it home. Nobel Peace Prize in Chemistry 2025 was awarded to Sasuma Kitagawa from Kyoto University in Japan, Professor Richard Robinson from the University of Melbourne, QS ranking.
Yeah.
Oh,
Melbourne.
hitting Melbourne. 18.
Yeah, it's something like
It's around
that.
there.
Yeah, I think it's
19. It's 19, I think.
I think it is 19,
Yeah,
yes.
yeah.
I was going to say 19.
Yeah.
Professor Omar Yagawi from the University of California, Berkeley. It is for the development of metal organic frameworks, or MOFs, that have been used to harvest water from desert air, capture carbon dioxide, store toxic gases, or catalyze chemical reactions. Essentially, MOFs are hybrid crystalline materials made up from positively charged metal ions linked together by linking molecules, and together they form a cage structure. If you imagine one of those balls that open and close in a toy shop, you know, those massive ones that can expand. Imagine that fully expanded.
Yeah.
Those are the linking molecules. That's their structure. They're hollow, so they have a huge internal surface area. The science has been 36 years in the making, when Robinson tested using an inherent property of atoms to combine positively charged copper ions to four-armed molecules. Upon combination, they bonded to create a well-formed crystal, but it was unstable and collapsed. Enter Kitagawa and Yagagi, who between 1992 and 2003 separately developed series of discoveries that built on Robinson's work. Kitagawa found that gases can flow in and out of the constructions made, and predicted that MOFs could be made flexible and could essentially trap and release methane, nitrogen, and oxygen without changing too much of their shape. Meanwhile, Yagi was building on the hype of MOFs from researchers and created at least 16 stable MOFs models that could be used to both expand and contract their kind of stable space structure. And in one instance, his research group harvested water from the desert air of Arizona. During the night, their MOFs material captured water vapor from the air, and when dawn came and the sun heated it, they were able to collect water. Through these three men's discoveries, chemists have been able to build tens of thousands of different MOF structures, opening up a world of opportunity to solve big chemistry challenges such as separating PFAS from water, capturing carbon dioxide, or even harvesting water from desert air. So this one has been a huge one in the making, much like a few of yours, Gabe. So I think this one has definitely been worth its prize.
I actually saw something else on this because I was really interested in the chemistry prize and the idea of MOFs, because it's essentially one of the things that you learn when you do chemistry is it's basically all about shapes.
Yes.
Everything's about shapes and how things fit together. And this is one of those things where it's a shape, holds itself up and can fill things can be put in it safely. One of those is carbon. But one of the other things they were thinking of is they think they can do this thing where essentially, we were talking earlier about with cancer drugs, it's about the delivery system, not what the drug is. They think they might be able to use MOFs as a delivery system because someone has designed one where essentially it breaks upon reaching an acid or a specific acidic environment, and they think they might be able to say like, hey, okay, we're going to pop these MOFs into your system with the encapsulated proteins or whatever we need to give you, and once they reach, I don't know biology, so forgive me with bodies, I don't understand human bodies, but let's say your spleen or something, I don't know if that's acidic, but if it is acidic, your spleen, and once it reaches that acidic environment, it breaks apart, delivers the drug, and it's very specific. It goes to the right spot. So that's another one of the really cool things these MOFs can do.
Cool.
I think that's it.
Cool. That's the wrap. No idea. Space news. SpaceX just flew their latest fully reusable rocket called Starship yesterday morning. That was Brisbane time. It was test 11, and it ticked all the boxes for a successful flight. It took off with all 33 Raptor 2 engines working. Hot stage separation was a success. The booster made it back to where it was meant to land in the Gulf. Starship deployed 8 mock satellites while in space using its Pez dispenser. It relit one of its 6 engines while in space. Then Starship performed some hard turns to wash off some of its speed and performed a textbook soft landing just off the coast of WA. Job done. The next test, Test 12, which will probably happen next year, will feature a new generation of Starship. It's version 3, which will be bigger and better. And for the record, the newly retired Starship, the version 2, had an overall success rate of around 40%. SpaceX will want version 3 to be a bit closer to 100% to realise NASA's moon obligations and to reach Mars before 2030. There was a flyby, apparently, of an asteroid. It's called Asteroid 2025, TF.
Oh. Cool.
It flew past Earth above the Antarctica on October 1, coming within 428 kilometres of Earth's surface.
Jesus. The penguins would have had a show.
Roughly the size of a small car. It was only detected after the fact. The European Space Agency later stated it posed no threat. That's good. I'm still amazed that it didn't take out some of our low Earth orbit satellites, which sit around that sort of... The ISS sits around that level of altitude. And I'll go through some terminology about asteroids. So an asteroid is a large rocky object, while a meteoroid is a much smaller one. The key difference is size. A meteoroid is generally considered to be less than 1 metre across and is often a fragment of an asteroid or comet. When a meteoroid enters Earth's atmosphere, it becomes a meteor or shooting star. And if it lands on the ground, it's called a meteorite.
And a crater.
Less than over? Yeah, and a crater.
The less you know.
The less you know.
Now, I've got a question / some trivia for you.
Oh, great.
Where would you likely find the longest continuously running computer?
Oh.
Wow.
Yeah.
That's a question because it depends on your definition of computer as well.
Yes,
Like,
it does.
are we talking about... Like, there's loads of, like, ancient computers that are not...
Are they still running, though?
Mechanical.
Yeah.
Continuously running.
Continuously running.
Continuously running.
Yeah.
I feel
I'm
like
going to say China or India.
I was going to say it's going to be in like some hospital somewhere because we all know those things are still running Windows 94.
That's so true.
That's so true.
Windows 95 has not been turned off since well before then.
And
It can't be
IE1.
turned off.
Yeah. Okay. So Max, tell us what the longest running one on record is, and then we'll see if anyone's got a better one to text in.
Well, as we know, Voyager 1 and its sister probe, Voyager 2, were both launched by NASA back in 1977. They are the first human objects to reach, human-built objects to reach interstellar space. And there is another record set by one of the twins. Voyager 2's computer command system has not been turned off since it first booted up 48 years ago on the 20th of August, 1977.
Oh my
making
God.
it the longest continuously running computer.
What about those ones in, like, air traffic control, though?
Surely
But they have to reboot them.
Does
This
it
has
get-
never
does
been
it-
rebooted.
Never
Does
been rebooted. Yeah,
That's
exactly. Never had a system update. Never had anything.
Is
It's
it
just-
entitled to long service leave?
At this point, surely.
Because even Voyager 1 had to reboot their system
my
to get it
God.
back online. So, yeah, but not Voyager 2. It's been pain-free, fault-free.
Damn.
Wow.
Good for them.
Anyway, that's it for No Idea this week. Sign us out, Gabe.
It is all we have time for this week. This Nobel-filled show was brought to you thanks to the Drunken Monkey Hypothesis and all of the subscribers to 4ZZZ. You can join them at 4ZZZ.org.au, where you can go through and look at all the different options to keep this station on air and support community radio and science radio. You can also, on that website, listen back to a bunch of the full recordings of this show with all the music, as well as a bunch of our previous shows. The condensed version is available on your podcasting platform of choice. To search up No Idea 4ZZZ. That's No with a K. No Idea. 5 stars still?
5 stars still, and getting stronger. You can also find us, just for funsies, you can find us on many social media platforms as well. If you want to see what we look like, break that wall.
I know that's a big if for radio.
It's a big if.
But
You
we're
can
on,
close your eyes.
We're on Instagram. We're on TikTok. We're on Facebook. No idea. 4ZZZ. We'll be back in your ears next week. Between now and then, keep an eye out for drunk chips. Put Coccolithophore Day in your 2026 calendar. Look up to checks for Asteroid 2025 TF before crossing the road. And lock up your moths.
Job done.
Nice.
And we'll speak to you next week.
See ya.
I'm a goddamn marvel of modern science.